Yeah, so I start the recording right now. Please note that you are being recorded. For the final exam, we will start discussing the details, and I will send those in multiple announcements and emails later this afternoon or tomorrow afternoon. The exam is going to be in person. The faculty has already sent out the information. I already sent you that out. And most probably it will be on Brightspace. So I will send those details out. Please make sure you have your computer ready and all that set up for that final exam. One note for those students who deferred the midterm, I haven't heard from anyone. Please reach out as soon as possible to rearrange the deferred exams. As for the assignment, there was, when we created this assignment, we had multiple groups that had issues. The TAs has reached out to some of you to reorganize, and we will make the final format. I have a meeting today and will send that email where you submit your assignment. Please don't submit your assignment at 11:59 on the deadline. Give yourself a few hours to do before. Assignments that will be submitted later, they will be like, you can read what's written in the syllabus. Okay. You can use some points if you submit the assignment late. Normally assignments are 2 weeks, 3 weeks. We did like almost a month for this assignment. For the assignment, we both submit. If you are in a group, you don't need both of you to submit. Only one submission is enough. If it happens that you didn't work in a group because you didn't have a partner or you were like just left out, then you submit on your own. But you don't have to submit 2 assignments per group. Only one is enough. Okay. So as for today, you could notice in the midterm that there were few parts of the last lecture were not fully covered in the exam. So screen. Let me know if you can see the screen. Oh, I'm sorry, this is not updated. Anyway, this is module 5. Okay. The title was not updated here. So again, the office hours are still as planned before, on Thursday from 12 to 1. Feel free to join me if you have any questions. So up till now, we covered almost lots of mechanisms that regulate the DNA the DNA replication, the transcription, translation into proteins. And now we are moving on to discussing how the cell uses the energy that it takes from the food and how it breaks it down into molecular units that can be utilized. If you remember from the last class, we have a couple of slides that are still in this lecture here that I would like to revisit so that I refresh your memory, because those 2 slides are in fact the summary slides for the end of this lecture. So today we are mostly going to look at the metabolism, define what is a metabolism, if it's a catabolic or anabolic reaction. We will explain a little bit about how ATP is generated. This is just refreshing for your memory. And then we will describe how the process of metabolism takes place in the mitochondria and how we generate ATP from those molecules. In the survey, some of you said you already have studied these. So some parts of those are not going to be like mandatory to memorize, like the names of the enzymes. Not all the names of the enzymes are mandatory to memorize. However, some of them are important, especially the one I will stress out during and I will highlight during the class. So, and finally, we will try to connect the metabolic dysfunction to some disease states. And as I mentioned before, the midterm, probably this is the last class where we discuss diseases from the perspective of molecular abundancy, or the mechanisms of how these molecules are being synthesized and processed. We will move from next lecture up discussing states of disease, where we describe a disease and we revert it back. We try to understand what is the molecular mechanism. So that will give you a different perspective of this course. Okay, and this lecture probably is going to be very heavy in terms of the names of the enzymes. So please bear with me a bit. I remember this was probably the last slide was included in the exam, where we discussed that the metabolic pathways play an important role when it comes to the cell homeostasis, and definitely when there's a cell injury or cell death. So metabolic pathways, as defined, these are cellular respiration, where the cell extracts energy from molecules. In other words, the cells would extract energy from the bonds of these molecules. And if you keep this notion in mind, it will be easy for you to understand that whenever you have a molecule like a glucose, which has like 6 carbons, it will be broken down into smaller molecules. And when these bonds are broken down, then the energy that is released can be used to generate a different form of energy that can be used in the cell. And therefore, the cell has to be prepared when this molecule is broken, or when other molecules like fatty acids or proteins are broken, then that energy that is released, some other molecules has to be ready to capture the electrons. And once those electrons are captured, this is how they are stored, because those electron carrier molecules will be able to deposit these electrons somewhere else, and we use the energy somewhere else. Okay, so in that lecture, we discussed the importance of the electron transport chain and how the ATP synthesis took place. And I just want to refresh your memory one more time with that video that probably it's at the end. Okay, so it's here. So based on this, we also classified what are the definition of electron carriers. And we stated that electron carriers are molecules that are able to have 2 different setups, one that is oxidized and one that is reduced. And we've discussed extensively the definition of an oxidizing agent versus a reducing agent, or an oxidized molecule versus a reduced molecule. And we link this to the source of these oxidized and reduced agents For example, the NAD, which is the nicotine adenine dinucleotide, the NAD, that comes from the vitamin B3 mostly. So this is linked to our diet directly. So the precursor for the NAD is from the vitamin B3. The other thing we discussed is that the oxidizing agents, when you have NAD with the plus sign, this means this is a molecule that is very well prepared to capture electrons. And when it performs this function or this duty, then the molecule beside, which is signed here as RH, which is a reducing agent, this molecule will become oxidized. And this oxidizing agent, once it gained that electron, it will be reduced. So you can see the NAD plus, it's oxidizing agent, it's an oxidizing agent. It's ready to capture electrons. So it's gonna be looking around where it can capture these electrons from. Therefore, it has a high affinity to capture those electrons. And it's looking for a reducing agent, which is the RH in this condition. Once the NAD plus capture those electrons, it will have one proton on top, and therefore now it's a reduced agent. So it's carrying enough electrons. And at the same time, this product here, this substrate here become oxidized. So this is the major concept, how the electron carriers work. The carriers that have the highest affinities will be placed in the electron transport chain in the mitochondria in a way where the lowest affinity comes with something a little bit higher affinity, then a higher affinity and a higher affinity, so that the electrons will keep going from the lowest affinity to the highest affinity till the last cascade of that electron transport chain. And we mentioned that we have more than one of those molecules, which is FADH, which works the same way. Okay, so here's the compounds, and you can clearly see that the difference is only that we have obtained one proton, one electron here, and that electron is enough to make this oxidized form of NAD plus into a reduced form with NADH. Okay, why we are discussing this today? Because in principle, we discussed before that the major currency of energy is in fact the ATP. So this is the second molecule that we discussed, the flavin adenine dinucleotide, which is FAD, and it comes again from the precursor of, it's the precursor of this is the vitamin B2. And when this is reduced, it will be FADH2. I would like again to stress that you should have watched this electron transport chain. It's very, very important for you to understand how the electron transport chain worked. And I want to stop a second and ask you, do you want me to refresh your memory showing you this video one more time? Because it's going to be so crucial to understand the slides coming. If you remember it, I will skip it and go forward with the slides. So I need your input. Okay, so let's take a few minutes to watch this video. Okay. Could you please let me know if you see my screen on the video? Okay. Okay, so let's watch this, and then we will go to the discussion of the glycolysis and the Krebs cycle after that.
All eukaryotic cells, from yeast to those that make up the human body, contain membrane-bound organelles with specialized functions. Mitochondria are double-membraned organelles that harness most of the energy that cells need to grow and reproduce. Nearly all of this energy comes from reactions that take place at the inner mitochondrial membrane. One of the key roles of this membrane is to act as a barrier to positively charged particles called protons, thus allowing a concentration gradient to be maintained where the intermembrane space has far more protons than the matrix.
I just want you to know, so that's where we're going to discuss most of the Krebs cycle today. So that's where you have lower amount of protons, and most of these protons are higher in the intermembrane space.
The membrane also contains a large protein complex called the F1-F0 ATP synthase, which uses the proton gradient to drive the synthesis of ATP molecules. These ATP molecules ultimately provide the energy for most of the cell's reactions. Just as man-made power plants produce electrical energy by using the flow of wind, water, or steam to rotate a turbine, the synthase makes ATP by using proton flow from one side of the inner membrane to the other to rotate protein subunits. If there is no proton gradient, synthase subunits stop rotating, and the cell can quickly become starved of energy and die. Therefore, the protein complexes and small molecules that establish this gradient and maintain it play an essential role in the life of the cell. At the heart of this system are four protein complexes, numbered 1 through 4. Complexes 1, 3, and 4 directly pump protons from the matrix into the intermembrane space. Complex 2 does not directly pump protons, but it does promote proton pumping in complexes 3 and 4. Proton pumping requires energy, and the four protein complexes get this energy by transferring electrons through a series of coupled reactions. This linked process of electron transport is why the four complexes are collectively referred to as the electron transport chain. Let's focus on complex 1. A byproduct of sugar metabolism called NADH deposits two high-energy electrons in complex 1, where they are passed along a chain of redox centers. Redox centers are clusters of atoms that have different affinities for electrons based on their unique atomic configurations. Let's closely consider a pair of redox centers to reveal two reasons why an electron moves from the top redox center to the bottom. First, the bottom redox center has higher affinity than the top one. Second, the distance between these adjacent redox centers is ideal for an electron jump to occur, which explains why electrons typically don't bypass the bottom redox center. A small amount of energy is released each time an electron is passed between redox centers. Complex 1 harnesses this energy across all the redox centers and uses it to pump protons. The last redox center in complex 1 donates 2 electrons to a coenzyme Q molecule. Complex 2 is similar to complex 1 in 2 important ways. First, high-energy electrons also enter complex 2 via a byproduct of sugar metabolism, although here the molecule is FADH2. Second, complex 2 also transfers electrons between several redox centers before donating them to coenzyme Q. One major difference, however, is that complex 2 does not use the energy liberated to pump protons. Coenzyme Q molecules from complexes 1 and 2 donate their electrons to complex 3. One electron is recyclable and can re-enter complex 3 later, but the other passes through 2 redox centers before reaching cytochrome C. Cytochrome C carries the electron to complex 4. The electron transport chain ends in complex 4, where a series of reactions involving 4 electrons converts a molecule of oxygen to 2 molecules of water. The proton gradient is strengthened because 4 protons from the matrix are incorporated into water molecules, and another 4 are pumped into the intermembrane space. In the absence of oxygen, the electron transfer comes to a halt, meaning that ATP synthesis also stops. Indeed, the reason we breathe oxygen is so that it can serve as the final electron acceptor at the end of the electron transport chain. In this animation, we have explored each protein complex in isolation, but in reality, they are very densely packed. Together, they effectively make the entire surface of the inner mitochondrial membrane a giant cellular power plant.
Okay, so now you could see how we use the NADH and the FADH2. It's also briefly mentioned how the electrons are transmitted from those complexes into complex 4 by the cytochrome C. And therefore, you could imagine if there is a change in the level of the expression of the cytochrome C, or if there is a release of cytochrome C from the inner membrane, this is a big problem for the electron transport chain. So this is one thing you have to keep in mind. The other thing is that what has been mentioned in the last step, where oxygen is going to accept those electrons at the end and make the formation of the water with the 4 protons. So you could imagine how the loss of oxygen will directly cause a block of the ATP synthesis, and therefore this is a big problem because the powerhouse of the cell is kind of stopping, and that's why the cell has to find quickly how to perform in conditions where there is very little oxygen. This will come into a practice when we discuss ischemia, and like in the brain, ischemia in the heart, particularly in these 2 tissues, because the loss of oxygen cannot be tolerated. And whenever there's an ischemia in the heart or in the brain, there will be a direct damage for a particular site of that tissue, which will practically cause the death of these 2 tissues. Why brain and heart? Because they are not regenerative, and therefore, if a cell dies, there's nothing that can replace it. And once it dies, the function of that cell is not going to be maintained. Okay, so now let's go back to discuss how we reach to generate the NADH and the FADH2 in the cell, starting from the food that we intake every day. Okay, so let me go back here. Just okay. So we already discussed that. And now remember, again, ATP is the energy currency, and it's the molecule by which we perform all this, the cell perform all this electron transport chain in order to maintain a difference in the H plus gradient, in the proton gradient. And you could see in the beginning of that video that the electron transport chain is using all that energy as FADH and FADH2 and NADH in order to pump those protons outside, which will then be used by the ATP synthase to turn on that turbine that was represented as a turbine in order to produce the ATP. So ATP synthesis is just that last step of the process, which later on we could use it as we described in multiple aspects. One of them was transcription elongation. We bring the ATP and the power, the energy that is stored in these phosphate bonds is going to be used for the process. One of the examples here is the transcription elongation. Okay, so I hope this refreshed your memory on what we have discussed before, and then we will move on to the oxidative phosphorylation. I don't remember if I described this slide before. There is also another video to describe for you mitochondrial diseases and how the mitochondrial damage cause effects on the matrix cristae and the membrane of the mitochondria. And it's very clear for you now that if there is any damage that is caused at the level of the membranes of the mitochondria, which causes a leakiness so that these electron transport chains are not very tight, then the energy is gonna not be produced in these mitochondria. At the end of this lecture, we will also describe how this is integrated into different cellular processes, like how it could affect the transcription, translation, DNA regulation, proteins. So we described, most probably we described this slide where the ATP synthesis process takes place, and most of the ATP is generated at the end of the electron transport chain. This is a process that we call oxidative phosphorylation. Now, for the energy that we generated, it could come from different sources. I'm sure you all know that the glucose is the easiest source of energy because carbohydrates in general are very easy to break down. However, lipids and fats are also a very important source of energy. And we will describe how we get the energy from those. So let's dive into the metabolic breakdown. So few definitions before we start, so we are on the same page. So metabolism in general is all these biochemical reactions that happen in the body. And therefore, it's the breakdown, as we said, of these molecules in order to produce the energy that the cellular functions can utilize. So anything we eat, from proteins, fats, carbohydrates, is hydrolyzed in the intestinal tract. And then, of course, we have multiple enzymes that are responsible for this process. At the end product would be amino acids, the basic units of proteins, fatty acids, the basic units of fats, and glucose, which is the basic unit of the carbohydrates. These will be absorbed and then will be incorporated into the cells, where we have to break those down in order to generate the ATP. So the energy releasing step is when you break those molecules into smaller pieces and release the energy of those bonds. This is called breaking catabolism. So breaking the molecules and therefore releasing the energy. In some aspects, we have to think in a reverse way, where probably we need to store that energy. And in this condition, we are speaking about using energy of metabolism in a way that we have to synthesize molecules. And that's called anabolism. Okay? This process generates the ATP, and this is one of the examples that we are discussing. Probably I will not describe everything related to proteins because for the lack of time, but I will provide you with some further reading if you are interested to study how an amino acid is converted into sugar or pyruvates, and it enters to produce energy. So this metabolic pathway, when we say a metabolic pathway, it's a series of reaction by which the end product of ATP generation takes place. Okay. As usual, I add for you the references from the book. Feel free to go and check more how the text if you need to read more about this. In addition, I will provide you with a couple of references that are really new from research papers as well. So the process of ATP synthesis, again, it's a metabolic pathway where we have a substrate, and now we have to discuss what type of substrate we will be using to generate the energy. Like this one molecule of glucose, and not necessarily I want you to memorize these numbers, but it's really important to sometimes keep some of the notions remembered, that one molecule of glucose has a 686 kilocalories. And the end product, when you have an ATP, the ATP will carry more or less like 7 kilocalories. So you're generating probably multiple of those ATP from the glucose. Then ATP is the currency where it stores the energy and transfers it. So how does it do that? And how do we reach to get to the point of ATP synthesis? So broadly, the catabolic pathways by which we break down molecules are divided into 3 levels. The first level is the glycolysis, by which we will break down the glucose into pyruvates, which is now glucose is a 6-carbon molecule, and pyruvate is the 3-carbon molecule. So you will break glucose into 2 pyruvates, and then you will generate in total 2 ATP from this process. And this process is in the cytoplasm. Now, the pyruvates, these molecules will be transported into the mitochondria. And it will enter to Krebs cycle or the TCA cycle or the citric acid cycle. There's like 3 different names for this cycle, where it will be converted into acetyl-CoA, acetyl coenzyme A, where This 3-carbon sugar, it enters into a multiple series of reactions to produce NADH and FADH2. So if you relate what we have discussed and what we have seen in the video, thinking about electron transport chain, electron carriers, and the redox centers. So the redox centers are sitting in the intermembrane space, inner membrane space. Then you have the glucose, which is in the cytoplasm. Then the pyruvates are generated. They are shipped into the inner, into the matrix of the mitochondria. And then these acetyl-CoA will generate the NADH and FADH2 in the matrix that will be used in the electron transport chain. So you got that molecule of glucose generating the NADH and FADH2 that will turn out the ATP synthesis in the mitochondrial inner membrane. Now, for the oxidative phosphorylation in the mitochondria, it's divided into 2 parts. One of them, like we have seen that in the video. The first part is the electron transport chain, where we have these 2 molecules circulate through the 4 complexes and then generate the ATP, mainly by the ATP synthase through the proton pump, through the ATP synthase F0, F1. Okay, so this is generally how one molecule generates an energy. But what makes one molecule produce less energy or more energy? It's depending on how many carbon it has. So let's look closer to the breakdown of the food as we digest it. So we have here, let me just move this one a little bit here. So we have here the first phase of catabolism, where proteins, lipids, and polysaccharides found in the food are first digested. Okay, and the digestion process is going to make these into their initial building blocks. As I said, fat will be broken to fatty acids, polysaccharides into sugars, monosaccharides, and the proteins as amino acids. So that's a digestion phase that happens across the organs. It happens in the intestine. It has been shipped within the blood, and then it has been filtrated in the liver. Then it will be available for the cells. Now, the second step is the glycolysis and the oxidation of that, which means glycolysis. Glycolysis is the breakdown of these sugars, as I mentioned before, into pyruvates. And amino acids can enter into this process because amino acids are like single blocks. And therefore, each type of amino acid depends on the complexity of the R group, could generate different levels of metabolites. Some of these will be directly feeding into the pyruvates. Some of those will be feeding to the acetyl coenzyme A, and some will be feeding directly to the Krebs cycle. And the end product will be that we deaminate those. We're gonna get rid of the amine group. So this is called deamination. And the end product, that's where urea comes from. Okay. If we look for the process of glycolysis, this is gonna be producing pyruvates, 3 carbon molecules, and that pyruvate is the one that will be joining the Krebs cycle as an acetyl-CoA. The same thing for the fatty acid. It's a little bit more complicated. I will be visiting this extensively in the last part of the lecture. Okay, so the digestion phase, we have those building blocks, amino acids, sugars, and fatty acids. The glycolysis and oxidation is where we produce ATP directly from the process of the glycolysis. And as you can see, we don't produce too much energy from the glycolysis. Whereas the Krebs cycle it's where most of the energy is being produced. The citric acid cycle or the citrate cycle or the tricarboxylic acid cycle or the Krebs cycle, based on the name of the scientists who described this process, it's how these molecules being circulating, donating their electrons, broken down, and generate the NADH molecules. So the last step is when these molecules enter to the Krebs cycle, these will produce what? NADH and FADH2. And that's where these two molecules perform the oxidative phosphorylation process where ATP is generated in the mitochondria. So I hope this gives you a whole picture from eating, breaking down these into subunits, and then generating molecules like pyruvate, acetyl coenzyme, and those will enter to a cycle where efficiently will produce a lot of NADH and FADH2, and then these reducing agents, NADH and FADH2, will be recruited into the electron transport chain that you've seen in the video, where you will use for the process of ATP synthesis. Okay? Of course, this is not always a clean process because there will be side products like carbon dioxide that can be excreted, and that's why we need to always get rid of the carbon dioxide. And this process also comes with the generation of the deamination of amino acids, which causes the generation of the urea as well. And finally, the process of ATP synthesis, as you saw, that oxygen, as meant in the end of the video, is going to accept the electrons and two protons to generate a molecule of water, and that water now can be excreted. So this is how the process from the beginning of eating a food and vitamins and mix of stuff that will become now the currency that the cell can use as ATP. It's not as easy as it looks like in this scheme, because if we look at, okay, what happens here? Wait, my computer just decided to freeze. Okay. So this is just to remind you that in phase 3, we generate NADH, FADH2. Maybe I repeated this multiple times now. And the side product would be the carbon dioxide. Now, NADH, FADH2 will transfer these electrons to the electron transport chain, and that's the video that we just watched. Okay, so let's start describing glycolysis. And then we will take a little break, and then we come back to describe Krebs cycle. And then we take a little break to come back and discuss why we are studying these two. So in glycolysis, this is a metabolic pathway. Of course, it's, as the name says, I always tell you, you have to really understand the notions, how these terms are made. Glycolysis, you can read it glycolysis or glycolysis. So you are lysing glucose, therefore you are breaking glucose down into molecules that in this case are called pyruvates. The free energy that is released from glucose is used to form ATP and NADH. Well, the first half of glycolysis, which is presented here for you in a series of 5 reactions, uses energy in order to phosphorylate glucose. So now you have a raw material, which is the glucose, which is a 6-carbon molecule, right? But in order to break down this molecule to get the energy from the bonds that are the energy that is stored within the bonds of the glucose, you have to first prepare it to be ready to release those molecules. Because the end product you want is this molecule here, which is called pyruvate. And in order to generate the pyruvate, first you have to add a phosphatic group, and this is a hexokinase. You can see from the name hexa, it's like 6, and kinase is enzyme that adds a phosphatic group. So the glucose is becoming glucose 6-phosphate. So the carbon number 6, because this carbon here, it has oxygen with double bond, is the number 1, and then this is the number 6. So you can read here glucose 6-phosphate. The phosphatic group has been added to the carbon number 6. And in order to do this process, the hexokinase, meaning the enzyme that adds the phosphate to the carbon number 6, needs energy. And that energy comes by spending energy first from an ATP to ADP. So you already lost one molecule. So someone has a question? Do we need to know this structure for the exam? That's a very good question. No, you don't need to. Not all of them, not all of them. Like, for example, pyruvate, yes. Now, probably you should not care about the structure itself, but I could present you this cascade, and I might ask you about a name of one of the enzymes. Okay? And again, I'm stressing on some of these enzymes, not all of them. So you would understand from how much I'm stressing on a certain enzyme that it's very important to know. Particularly in this is the step 1 and the step 3, because we are losing the energy here. Because we break down an ATP into ADP and then ATP to ADP. I'm not going to ask you if your question was, write me the difference between glucose and glucose 6-phosphate. I'm not going to ask you to this question in terms of the carbon oxygen phosphate. Okay, now you will see a lot of changes in the metabolic state or in the molecular state by using enzymes that are isomerases. This is one of the key aspects of metabolism, where, you know, isomerases, they just change the topography of the molecule, but they don't really change the number of the molecules of carbon and oxygen. So for example, if you look here from glucose 6-phosphate to fructose 6-phosphate. So what happened? If you look at the molecule, you will see that nothing happened at the level of carbons. This is 1, 2, 3, 4, 5, and 6. They are exactly the same. Nothing had changed. But the phosphoglucose isomerase, what it does is the switch between the carbon 1 and carbon 2, where the double bond with the oxygen in carbon number 1 here. Now the isomerase switched that between carbon 1 and carbon 2. And therefore now you have exact same number of carbons, exact same number of oxygens and phosphates, but now the molecule has a different topology. Okay? So, and this is needed because most probably the process cannot continue to reach the pyruvate without doing these intermediate stages. So now you got glucose 6-phosphate and fructose 6-phosphate with exact same number of molecules with just a change in the organization and topology. Now, the next step is, again, to lose one ATP molecule. And you can see here, losing one ATP molecule was with a kinase enzyme, because the kinase has the capability to steal that phosphate and add it there. And again, the name is just indicative of what's going on. This is a hexokinase. It added the phosphate to this carbon number 6. And here is a phosphofructokinase, because now we are dealing with the fructose. And now it will take that phosphate group and it added on the other side. So you could understand that the process before, this oxygen is not going to accept any more electrons. So it was important to move it in from carbon 1 to carbon 2 in the phosphofructose. Then now we have an OH. You can easily lose that H group and get a bond with the phosphate. And that's exactly what happened. So now you have fructose, but now on carbon number 1 and carbon number 6, you have phosphate. And since you have 2, you will call it fructose 1 and 6 biphosphate, means 2 phosphates. Okay? The next step would be is the breakdown of these molecules into 2. So if we break down this molecule into 2, we will have the dihydroxyacetone phosphate, and we will have the glyceraldehyde 3-phosphate. Like, don't tell me why they chose these names. I'm not going to go into that, okay? But we have a 3 carbon units with a phosphate group on one side, where the oxygen is on the second carbon, and that's why it's a dihydroxyacetone phosphate. And on the other side, you have the carbon number 1, which has the double bond with the oxygen. So this is a glyceraldehyde 3-phosphate. The most important for you is to understand now that this 6 carbon glucose molecule is in fact broken down by this enzyme into 3 carbon molecules. And each one of those has a phosphate group. So that's still now step 5. Next, what happens to these 2 molecules? Okay. So in the second half of the glycolysis, it involves phosphorylation without ATP investment. So in the first step, we lost already 2 ATPs. But now we are not going to use any more ATPs. Now we have the energy that comes from breaking down the 6 molecules, the 6 carbon molecule into 2 3 carbon molecule. And now we have the 2 NADH and 4 ATP that will be synthesized from this process. So if you look at the net gain, you have produced 4 ATP, but you have lost 2 in the first step. And then the total amount of ATP you generate from one glucose molecule is only 2 ATPs. Okay? So now we will continue with the glyceraldehyde 3-phosphate. The glyceraldehyde 3-phosphate has this high energy bond with the phosphate group. So what you can do is generating an NADH by releasing the energy that is present with this molecule. Okay, so you will be, I'm sorry, this I jumped in the process. So in principle, you will have the NAD plus that will bring one more of these inorganic phosphates, so that this molecule become diphosphorylated again. So you can see now it's the glyceraldehyde 3-phosphate that will become 1 and 3 at carbon 1 and at carbon 3 by 2 phosphoglycerate. Phosphoglycerate. Okay? So now we have 2 phosphatic groups. The next step is to release the energy that the phosphate bond has. So this will be the first step where we take back one of the ATPs that we used by removing this phosphate and gaining energy to generate the ATP. And of course, these, I'm not going to ask you about the names. These are like really crazy to remember. The only thing I want you to remember, as I said, is some of the enzymes that I mentioned and some of the, particularly the steps where the ATP and ATP are being generated. Okay? So, but you can see that now that it's written here 2x. So we will describe why it's 2x, because if we go back in the steps, we have two molecules that are 3 carbon, and we just continue discussing about one of them, okay? That means this molecule, the glyceraldehyde 3-phosphate, is ready to continue in the cascade of the process. And in other words, we have to work a little bit more on the dihydroacetone phosphate to become ready to use it in the cascade of 6, 7, and 8, and 9, and 10. Okay, so if we continue with this one, we see that we first released one of the phosphates from the carbon here, carbon number 1. So you see now the 1,3 by bisphosphate phosphoglycerate become only 3 phosphoglycerate. And the next step is, as I said, always there's some sort of reorganization. So now you just switch that phosphate group from carbon number 3 into carbon number 2. And you can see the name didn't change much. 2-phosphoglycerate. So now the phosphate is on carbon number 2. This is done by phosphoglycerate mutase, and most probably this is an isomerase. Again, because when a molecule has the same amount, the same number of components like carbon, oxygen, and phosphate, and it's just the reorganization was changed, this is most probably an isomerase that performed the function. And at this last step, in the presence of oxygen, that's again important, in the presence of oxygen, water will be generated, and we will get this phosphophenol pyruvate. Now, this phosphophenol pyruvate is the end product where we get out one more ATP and generate that pyruvate molecule. This was extensive, I know. I don't know if you have studied it before. I hope I give you a good overview of how one molecule of glucose go through the process. Now, definitely there's the name a lot of enzymes that's very difficult to remember. All I want you to care mostly for the steps where the ATP is produced or broken down. Okay, now just let me finish one notion of how the second part, which is the dihydroxyacetone phosphate, is reused, because we continue only with a 3 carbon, and we produced, as seen in this scheme, 1 ATP, 2 ATP, and in the previous slide we lost 2 ATP. So till now we have 0 gain in terms of ATP number, but we produced NADH one time. Okay, so the second part comes from the usage of the dihydroxyacetone, where it is converted, and this enzyme is important to remember, it is converted to a glyceraldehyde 3-phosphate by the enzyme triose phosphate isomerase. So again, remember, when you reorganize that molecule, you use an isomerase. And this is a reversible reaction, depends on how much we have. It can be converted forward or reverse. So what happens is that when we get to the point where we have the fructose 1,6-diphosphate in the step number 3, and then we break it down into these 2 3-carbon molecules, this enzyme is going to take care of converting the dihydroxyacetone, okay, into the glyceraldehyde. So it takes one more step to do that. While this glyceraldehyde is ready to directly go through the process, this second 3-carbon molecule needs this enzyme to prepare it back to become glyceraldehyde 3-phosphate. And then that's why you will have 2x from one glucose, that one comes from the glyceraldehyde itself and one that comes from the dihydroxyacetone that will be converted to glyceraldehyde. And that's why you have 2x in that previous slide. And this explains why we produce 2 times ATP. So in general, you are producing 4 ATPs as written here, and we lost 2 in the previous step, so we have the net of 2. So here is a video for you as well to watch and see how these processes take place, and it's good to give you some visual memory about the process of the glycolysis. Okay, one last thing I want to mention is, okay, we produced pyruvates. And now that pyruvate, how can we use it in the Krebs cycle? So the first step is we have to think about that this pyruvate is the first precursor for generating NADH. Okay, and if you see here, the major, like it's like 3 different routes. If we focus right now on this route, where in conditions where the cell has a nutrient and oxygen, then this pyruvate will aid in the production of the NADH in the process where it's being converted to acetyl-CoA. Okay? And then acetyl-CoA is going to be the currency that we go to the Krebs cycle. Now, in the absence of the oxygen, then pyruvate cannot produce the NADH. On the contrary, you can see that you will reuse the NADH in order to break down the pyruvate into lactate or lactic acid. Therefore, this is an energy demanding step. So the presence of oxygen is very, very important. Even if the molecule has been broken down into the currency that we need in this condition, which is pyruvate, it cannot go to acetyl coenzyme and Krebs cycle in absence of oxygen. Therefore, the process of the, for example, lactic acid accumulation in the cells, it happens when your body needs oxygen that you are not able to provide. And this happens, for example, if you have never trained since 6 months and then you decide you want to go to run. Therefore, your body is not prepared. And when you start running for the first 10 minutes, you are able to give the oxygen that is enough for your muscles to produce the energy. But later on, you are deciding to run, but your body is not getting the enough oxygen. So it starts to produce energy in a fermentation-like a process where your pyruvates most probably are going to be converted into lactic acid. This process will cause the lactic acid to be accumulated in your muscles, and that's what will give you a little bit pain 2-3 days after your first training. Okay, there is another condition where these pyruvates, in the absence of the oxygen, will be transformed into the acetaldehydes by the loss of the carbon dioxide, and therefore the generation of the ethanol. And in this condition is more happening in yeast. Again, this is an energy demanding step. So your best bet is to break down the glucose, generate the pyruvates, and then that pyruvate, in the presence of oxygen, go into the acetyl coenzyme and the Krebs cycle, which we will describe in the next part of this lecture. Okay, now we're going to take a break till 10, and we come back at 10 to describe the Krebs cycle and the mitochondrial damage. I will be staying here for the next few minutes. If you have any questions, please stay and raise those questions. I will pause the recording during this break. Okay. Okay, so we described the glycolysis, and then we ended up with having the generation of the pyruvates. These pyruvates will enter the Krebs cycle. This is depending on the presence of oxygen. Again, there's further reading for you guys here about the anaerobic glycolysis, which might happen in the absence of glucose. Now, we will start describing how glucose that is now in the form of the pyruvate has to be transported from the cytosol to the mitochondria. Excuse me. And then pyruvate, once it's in the mitochondrial matrix, where it will go to the oxidative phosphorylation. Okay, please read this slide on your own, just because I kind of touched on what happens in the absence of the glucose for the pyruvates. So the next part is the tricarboxylic acid cycle or Krebs cycle. So in aerobic conditions where pyruvate is available, oxygen is available, pyruvate is primarily transported into the matrix. And this is involved in two different processes. The first is a voltage-dependent anion channel, which is called the VDAC, which is on the outer membrane of the mitochondria. And the second one is the mitochondrial pyruvate carrier, which MPC, which is on the inner membrane. So this pyruvate molecule will has to pass through these two gated channels in order to become available in the mitochondrial matrix. Okay, now this process has to be accompanied by three different processes. One, it will have a byproduct of carbon dioxide. Two, it will be generating the first NADH molecule, and this is one of the molecules we need for the transport carriers. And third is to merge the pyruvate in the form of an acetyl-CoA by having acetyl-CoA bounded to that molecule. So all the time you have to bear in mind that you have to count the carbon numbers that are being in that molecule. Remember, glucose was 6 and pyruvates are 3, okay? Upon entry through these two channels, what will happen? You already have a carbon dioxide that is being generated. This carbon dioxide, this means you lost one of the carbons. So you will end up with only 2 carbon molecule. And now this 2 carbon molecule is bound by the coenzyme A. The coenzyme A had this sulfur group. Okay, so the name of this molecule is acetyl coenzyme because this is an acetyl group and that's the coenzyme group. And the bond between those is by the thiol or the sulfur molecule. Okay, I wrote these details down that these are converted to acetyl coenzyme, acetyl-CoA, and a carbon dioxide. So you have it here, acetyl coenzyme and carbon dioxide by the enzyme pyruvate dehydrogenase complex. Okay, so there's the pyruvate dehydrogenase complex. This is the one complex that deals with generating the acetyl-CoA from the pyruvate and releasing a carbon dioxide. And again, remember that you are breaking down a 3 carbon molecule into a 2 carbon molecule. The energy that you have lost, not lost, that you have released, is the one that you're going to use for the generation of the NADH. And now we achieved the first NADH just by the entry of the molecule. For access to the mitochondrial matrix, 3 layers must have been traversed: the outer molecule, the intermembrane space, and the mitochondria. This is what I mentioned in the previous slide. And most of the metabolic activities are taking place in the matrix, which is the inner part of the mitochondria. Okay, more reading references are always listed on those slides. Okay, now we have the pyruvate in the mitochondria, and now it's in a different format, which is the acetyl-CoA. So just for you to remember what is acetyl-CoA, this is a key cofactor, and it is made up of two things that you already know very well, two components that you know very well right now, which is the cysteine, which is here an amino acid compound, in addition to adenosine, and then this molecule there that connects the two. Okay, so the CoA, the coenzyme has the thiol group, and the thiol group is is the part that is mediating or making the reaction much easier with different molecules. So coenzyme A is a critical cofactor in fatty acid metabolism as well. So what we have seen in this previous slide, where this pyruvate becomes acetyl-CoA, it will be a similar process where fatty acids perform the same. It's a little bit more complicated, but it's the same concept. So when you say acetyl coenzyme A, this is an acetyl group with a CoA, so that's how it looks like. So this is the 2 carbons, this is the sulfur, and the CoA, which is this part here, the coenzyme A. So this is a key central metabolic molecule, and you will see that exactly as ATP is the energy currency, this is the intermediate where most of our food is broken down to become acetyl-CoA that we can use in the energy processing. Okay, so the Krebs cycle. The Krebs cycle is a very complicated process, but let's, like, before going to the slide and go to the details, it's a very, very busy slide. The concepts, what happens in the Krebs cycle? So you got the pyruvate, you convert it into 2 carbon atoms, which is the acetyl-CoA. The idea why it's a cycle, it's because you have to start with oxaloacetate, and you go through the process all the way, and you will have oxaloacetate recovered. Now, oxaloacetate is a 4 carbon atom. So what do? We got here, let's merge these 2 slides together. So we got the pyruvate, acetyl-CoA is generated, 2 carbon atoms. Let's go to that slide. It's represented in a different way. So you got the pyruvate, 3 carbon atoms, and you got the acetyl-CoA, the sulfur, the CoA. You generated a 2 carbon molecule, acetyl-CoA, you release the carbon dioxide, and you produce the first NADH. Okay? Now, this molecule is able to enter the citrate cycle. In order to enter the citrate cycle, we will use this 2 carbon molecule with the oxaloacetate, which is a 6 carbon molecule. Now, the Krebs cycle, or the tricarboxyl acid cycle, is also the citric acid cycle, because citrate is such a central molecule in this process. The first step is to get this oxaloacetate, and if you look at it, it's a 4 carbon molecule plus the acetyl-CoA, which is a 2 carbon molecule, to generate the first 6 carbon molecule, which is a citrate. Okay, You have to remember that only one enzyme, which is the succinate dehydrogenase, is bound to the inner membrane of the mitochondria. Only this molecule here, okay? The rest of the enzymes are in the matrix of the mitochondria. Therefore, the citric acid cycle has to somehow get those products, the succinate, into the membrane where this succinate dehydrogenase perform the formation of fumarate from the succinate. Okay, so what is written here is just telling you the first two steps, where 2-carbon acetyl group with a 4-carbon oxaloacetate make the 6-carbon citrate molecule, which is here. Okay, then during this, the citrate molecule is decreased in length 1 carbon at a time. What does it mean? It's similar to what we have seen with glucose. You broke a glucose into 3 carbon molecules, but you have only 2. Now, Krebs cycle is more efficient because we will have this 6-carbon molecule, and we will start breaking it down 1 carbon at a time. And each time we break 1 carbon, then we will be generating energy from that. Okay? So it says here, I will read it again for you. During this cycle, the citrate molecule is decreased in chain length 1 carbon at a time, regenerating at the end of the cycle the 4-carbon oxaloacetate molecule. So the 4-carbon oxaloacetate that we had started with, CoA, after going through all the steps, it will be regenerated. And now you will have more pyruvate coming to become acetyl-CoA that will find oxaloacetate, and then we go another cycle and another cycle and another cycle. And that's why Krebs cycle produces much more energy compared to glycolysis. Okay? Is it clear till now? Or is there anything you want me to go back again and describe? Okay, nothing shows up on my screen, so I'll go to the next slide. Now, it's the 2-carbon removed during the TCA cycle, which are not the same one that brought in with the acetyl group, that are completely oxidized to carbon dioxide. It's important that we are recycling the new carbons. So it's telling you here that the 2-carbon removed during the TCA cycle are not the same ones that we brought from outside. In other words, we are getting 2 fresh acetyl-CoAs and the oxaloacetate, and then we're gonna lose these during the process from 4 plus 2 to become 6. These 2 are new and these 4 are old carbons. I just want to say old and new to differentiate the 2. Now, when we go through the process and we come back to oxaloacetate, the 2 freshly added carbons are going to be staying here, and the other 2 that we lose them as carbon dioxide, 1 here and 1 there, most probably will be one of these 2. Okay? So during the TCA cycle, there are 4 reactions by which these electrons are transferred from the substrate, which is the electron accepting coenzyme. And that's the concept we described before. It's exact same thing here, where we will have NAD plus as an oxidizing agent waiting to the breakdown of this citrate to the right moment to become NAD plus one time, NAD plus another time, and NAD plus a third time. Okay, so you have 3 of those as NADH and 1 as FADH, which happen to be within this enzyme here, the succinyl dehydrogenase. So in principle, Each round is going to produce 4 of these electron carriers. So in terms of the steps, if we just make it easier for you, I wrote down each step, what happens in it, but the concepts are the same. Probably I want to go back on this because it's easier to describe as the carbon molecules are being changed. So again, you have a 6 carbon molecule. You don't do much here. You just reorganize it. And then the reorganization will bring it into a level that is much easier now to break down and gain the energy. So you go from citrate to aconate, sorry, and then to isocitrate. This is a very intermediate step. So from citrate to isocitrate, now you can generate the first NAD plus. If you look, if you generate that energy, this means you already broke down 1 carbon. That carbon is going to generate the first carbon dioxide. So you generate the energy and the carbon dioxide. You ended up with a 5 carbon molecule. This 5 carbon molecule, in the presence of the CoA, again, you will have the next step in generating the NADH. Another carbon is being produced, and now the molecule is associated again with the acetyl-CoA. Not the acetyl-CoA, the coenzyme A, sorry. You lost the second carbon, you produced energy, and now you will have the 4 carbon molecule. From this step down, it will be mostly generating ATP in a manner that doesn't require the breakdown of the carbons, because you can see this succinate, the alpha ketoglycerate here is a 4 carbon, the succinate is a 4 carbon, the fumarate is a 4 carbon, the malate is a 4 carbon, and the oxaloacetate is a 4 carbon. What is being changed is either you will have inorganic phosphate in the presence of this alpha ketoglycerate that will generate succinate, and therefore it takes the energy from this phosphate plus the CoA to generate the GTP, which is an energy molecule, or you will reorganize the molecule by isomerases, and therefore you get back the energy as you're going. So here, most of these steps from the succinate all the way to the oxaloacetate, you see that we are producing only 1 molecule of FADH2 plus at this point here, and the rest would be just to recycle the oxaloacetate back to the to that original step. Okay, so the steps are written here extensively, and I do have for you a video that will clearly describe the process. So let me know if you can see my screen, please, because it will describe for you these steps. Are you able to see the screen or the citric acid cycle? Yes, no, anybody? Yes. Yes, okay. So let's go.
We know by now that glycolysis takes place in the cytoplasm and leads to the formation of 2 molecules of pyruvate per molecule of glucose. These pyruvate molecules are converted into acetyl coenzyme A, also known as acetyl-CoA, and this happens inside the mitochondrial matrix. Therefore, acetyl-CoA is the key metabolic intermediate that links glycolysis and the citric acid cycle. For clarity, we will not show the coenzyme portion of acetyl-CoA, and instead are simply representing it as the 2 carbon molecule acetate. So how does the pyruvate get into the mitochondrial matrix? This depends on a carrier protein that transports pyruvate into the mitochondrial matrix, where there it is oxidized to form acetyl-CoA by the enzyme pyruvate dehydrogenase.
So this is very important to know.
In this reaction, a molecule of carbon dioxide is generated as pyruvate is oxidized to acetyl-CoA, and a molecule of NAD+ is reduced to NADH. This last reaction is not shown for clarity. The citric acid cycle consists of the following 8 steps and also harvests chemical energy, producing a total of 3 NADH molecules. In the first step of the citric acid cycle, the enzyme citrate synthase combines the 2-carbon acetyl group from acetyl-CoA with the 4-carbon molecule oxaloacetate. This generates the 6-carbon molecule citrate. In the second step, citrate is isomerized into isocitrate by aconitase. In the third step, isocitrate is oxidized by isocitrate dehydrogenase, generating the 5-carbon molecule alpha-ketoglutarate. In the process, 1 carbon dioxide molecule is released, and 1 NAD+ molecule is reduced to NADH. In the fourth step, alpha-ketoglutarate is decarboxylated and oxidized to produce another molecule of carbon dioxide by alpha-ketoglutarate dehydrogenase. The resulting molecule is combined with coenzyme A, forming succinyl-CoA. In addition, another molecule of NAD+ is reduced to form NADH. Note that in this diagram, we are showing the product of this reaction as succinate and are omitting the coenzyme A for clarity. In the fifth step, succinyl coenzyme A is converted into succinate by succinyl-CoA synthetase, releasing coenzyme A. This reaction also produces 1 molecule of GTP, another high-energy molecule, which is omitted from this diagram for clarity. In the sixth step, succinate is oxidized to fumarate by succinate dehydrogenase. Unlike all the other enzymes in the citric acid cycle, succinate dehydrogenase is embedded in the inner mitochondrial membrane and is also a component of the electron transport chain, in which it is also called complex 2. As complex 2 oxidizes succinate, it receives 2 electrons that it will ultimately pass on to coenzyme Q, which in turn will deliver its electrons to complex 3. Another interesting aspect of complex 2 is that it uses FAD rather than NAD+ to accept electrons. In the seventh step, fumarate is converted to malate by the enzyme fumarase. And finally, in the eighth step, the enzyme malate dehydrogenase oxidizes malate to reform oxaloacetate and reduces a molecule of NAD+ to NADH in the process. This regenerated oxaloacetate can then combine with another acetyl-CoA in the next cycle.
Okay. Let's stop a moment here if you have any questions before I go forward. Anything was not clear about this process? Matteo, we're not discussing about the exam now. I'm discussing about Krebs cycle. Most probably not all the exam is multiple choice. Some of the questions are not going to be multiple choice. Okay. Sounds good. So we have the end products of generating 3 NADH and 1 FADH in addition to 1 GTP. And now you can relate how the generation of the energy from the glucose into pyruvate into acetyl-CoA entered to the mitochondria and generated those electron carriers being reduced. Now remember what we have seen in the video in the beginning. The process of these molecules, NADH and FADH2, will be donating their electrons to the electron transport chain. The complex is 1, 2, 3, 4. and therefore pushing the process of ATP synthesis by the ATP synthase. And that's mostly by making a proton concentration much higher outside of the cell, outside of the matrix compared to the inner part of the mitochondria. Okay, so let me just get this back. Skip it. Okay. So the steps that you have seen in the video are written here in case you missed anything. You have to be careful that the succinate dehydrogenase is the only enzyme that is within the complex in the inner membrane of the mitochondria. And this representation is a little bit more like simpler than the previous one, which is showing you like the basic molecules. Now it says glucose or fatty acids, and in both situations, we have to get to the acetyl-CoA. Now, fatty acids, we will describe it in the next couple of slides, where it's a little bit more complicated to get to the level of the acetyl coenzyme A. So let's remember what we have described in the previous modules. So these couple of slides, we have shown how carbon, hydrogen, oxygen, and other molecules would play a role in generating these biomolecules. And one of those is the fatty acids. And the most important characteristics of fatty acids is this long chain of carbons, 1, 2, 3, 4, all the way to 12. And we described at that time that saturated fatty acid compared to the unsaturated fatty acid is mostly due to the having the double bonds compared to the single bonds between these carbon atoms. Now, when you look at what happens in terms of the glucose, we had a 6 carbon molecule, it becomes 3. Now, with the fatty acids, you can tell that we have much more carbons that we can use. And this is primarily why the fatty acids are a better source of energy, or like produce much more energy compared to a glucose molecule. Because if you think that the building block of the acetyl-CoA is only 2 carbon atoms plus the coenzyme A, then glucose is going to provide us with only 2 of these acetyl coenzyme A molecules. While in conditions of the fatty acids, then you will have each of these 2 carbon atoms can be broken down and therefore generate the acetyl-CoA. So per se, from here, you can say we can produce 12 divided by 2, like 6 acetyl-CoA. And these 6 acetyl-CoA, if you go back and multiply by 3 NADH and 1 FADH and 1 ATP, then you will have 5 times more of energy from one fatty acid compared to one glucose molecule. So also between saturated and unsaturated fatty acid, the processing is a little bit more complex. I'm not going to go to the difference between saturated versus unsaturated. It's already a very complex process at this point. I will just describe the generalities of the saturated fatty acids. So remember, the fatty acids play important roles in the membranes. They are components of the membranes, and they are when they form the phospholipids. But also when we eat those fats and we break them into fatty acids, then we have to process those into the acetyl-CoA. And this is a process that needs multiple levels of control. The first one would be is that when we call fatty acid oxidation, you always say it's a like you always hear that it's written like it's a beta oxidation. And this is related to the site of the cleavage of that fatty acid. I will be describing this in the next couple of slides as well. So let's walk through how the fatty acids, after being like, remember this slide here where we described that food is broken down into simple sugars, glucose, and now we described glycolysis, pyruvate, acetyl-CoA, all the way to NADH. And you know already what happens below by ATP synthesis. Proteins are much simpler. I'm not going to describe it. I will go to the fatty acids. So fats are now broken down into simple units of fatty acids. And now the fatty acids will feed into acetyl-CoA. So there's no intermediate between fatty acids and acetyl-CoA, while in glycolysis you have the pyruvate intermediate. So what happens when fatty acids are being hydrolyzed? Okay. Now, to be oxidized, or in other words, releasing the energy from those fatty acids, they first has to be transported through the cytoplasm to a coenzyme A, then move to the mitochondria. And be careful that now the name looks like similar to the acetyl-CoA. Don't get confused because fatty acids are broken down into 2 carbon unit acetyl-CoA, which we have described extensively till now. But the transport of the fats from the cytosol to the matrix of the mitochondria, this is another whole process. And this requires the generation of what we call acyl-CoA. Acyl-CoA, this means a fatty acid that is attached to the CoA molecule. So you are talking about, when you say acyl-CoA in red, it is in fact a fatty acid, which is one of these 3 guys, plus the coenzyme A, this one here. So that's an acyl-CoA, not acetyl-CoA. There's a big difference between the two. Okay, now the acyl-CoA is a molecule that's formed by attaching the acyl group, typically which in a fatty acid, to the coenzyme A. Okay. Why we're doing this? Because in order to cross the membranes, these fatty acids has to pass through a particular barrier, which is in this case called acylcarnitine. And there are 2 of them here, the carnitine palmitol transferase 1 and 2, which plays a role in the transport of the acylcarnitine to the inner membrane space. So this is a laborious step where you have the fatty acids broken down into simple fatty acids here. Then you got the CoA that is bound to the side chain of that fatty acid to generate the acyl-CoA. Then the acyl-CoA goes to the mitochondrial membrane where it finds where carnitine is to generate acylcarnitine. So in other words, a generation of combine of acyl-CoA and carnitine. Then acylcarnitine will be transported into the mitochondrial inner space to the matrix. And now you have the 2 components are separated again, where acylcarnitine is where carnitine is recycled, therefore it goes back and it can be recycled for another round. And the acyl-CoA, which is a very similar to the in concept, it's very similar to the acetyl-CoA, but it's much longer. It's now in the matrix of the mitochondria. But now it's not ready yet. It's still not ready yet because the acetyl-CoA, which is the 2 carbon molecule, is much, much smaller than the acyl-CoA. So what we need to do is to break down this into multiple 2 carbon compound to generate this one. So in other words, we're going to break this one to generate this one here. Okay. It's written here for you. The carotene complex is transported across the inner membrane of the mitochondria, after which the fatty acid is reattached to the coenzyme A in the mitochondrial matrix. So now we have it as acyl-CoA here. So what happens next? Then the process of the beta oxidation takes place. So here you have the acyl-CoA, which is the fatty acid with the side chain 1, 2, 3, and it's written R. This means could be like 5, 10, 15, whatever this fatty acid is. But it's attached to the fatty acid at the carboxyl group. Okay? So you have the CoA with the sulfur group, and then you have the carboxyl group. And then you would have the alpha and the beta carbons. So this is the carboxyl, this is alpha, and this is beta, and you can call it also the carboxyl group and 1 and 2. So in principle, this molecule here will have to pass through the beta oxidation process, which is multiple steps, to be broken down just at that carbon number 2. So if you look at this one here, you have the CoA group, the carboxyl group, carbon 1, carbon 2. So that's the acyl-CoA, that's a fatty acid. Now, when it passes through the beta oxidation, it will generate a couple of reduced molecules, FADH2 and NADH+. But then at the end of that process, the molecule will be ready to be cleaved, where you have 2 carbons and the CoA, which eventually we call acetyl-CoA, which is very similar to glucose right now. Glucose got pyruvate, acetyl-CoA, fatty acid. We go to acyl-CoA and then acetyl-CoA. So now this building molecule can go into the citrate cycle exactly as we just described. Any questions? Anybody is trying to ask a question? Okay. Could you please mute yourself if you're not asking a question? Okay. So as written here, the fatty acid group is transported according to the membrane with a carrier, and then in the mitochondria, the fatty acid molecule undergo stepwise disassembly, which means you have now the TCA cycle to generate a lot of energy as 3 NADH, 1 FADH2, and 1 ATP from each acyl group. But on top of this, as much as we have carbons there, as we can break down 2 molecules and go in this process to generate acetyl-CoA. So the more side chains we have, the more acetyl-CoA we'll have, and the more energy we'll be producing. And this explains why fatty acid would produce much more energy compared to glucose. And it explains why some organs like the heart would primarily depend on the generation of the energy on beta oxidation of fatty acids, not on glucose. And when the heart go to a diseased state, then the source of energy is going to be the okay, guys, somebody was talking, so I'm just muting everyone. Please make sure you're mute unless it's related to the lecture. Okay. Sounds good. So any questions regarding fatty acid oxidation or beta oxidation? I know this lecture is very heavy, but I promise that this will be probably the last molecular description and we will move to a little bit cell biology, and it will be much easier then. No questions. Okay, so I will now minimize this. And we go to watch this video together about beta oxidation. Okay, good.
I'm partnering with Disney to tell you about Disney Plus. Okay, top Disney Plus pick tonight. Secret Lives of Warm Wives season 2. I rewatched season 1 with you, remember?
Now, to break down fatty acids, we go through a process called beta oxidation. So I want to step us through that. The enzymes in the enzymes for beta oxidation are found in mitochondria and little organelles called peroxisomes. This process of breaking down fat generates more ATP per carbon than sugars do. Now, sugar metabolism is very important, and sugar metabolism is occurring more readily than fatty acid metabolism is, partly because sugar can travel in the bloodstream more easily than fat and fatty acids can. Fatty acid metabolism works in a breakdown process that proceeds by a chopping off of 2 carbons at a time from a fatty acid. And you can see that happening in this process here. On the top, I have a schematic representation of a fatty acid, and I've marked the 2 carbon pieces that get chopped off. We see, first of all, the release of the fatty acid, and the fatty acid is broken between what are called the alpha carbon, as you can see here, and the beta carbon here. So in the numbering scheme that's here, that's between carbons numbers 2 and 3. So either term is commonly used. That breakdown process, as I said, releases a single acetyl-CoA, that's a 2 carbon piece, and then continues along at 2 carbons at a time until the entire fatty acid has been broken down. Now, fatty acid oxidation is a multi-step process like most metabolic pathways, but there has to be some processing of the fatty acids to get them ready for the oxidation. Fatty acids that start in the cytoplasm have to first be activated and then moved into the mitochondria. But as we will see, that process is a little odd. The activation of a fatty acid begins in the cytoplasm, and this involves the attachment to the fatty acid of a molecule called coenzyme A, or as people more commonly say, CoA. This is an energy requiring step, and you can see that ATP energy is needed to do this, and the product of this is not ADP, as we've seen many times before, but actually AMP. And that's a reflection of the fact that this is a pretty high energy bond that's being created. The result of this action is a molecule called fatty acyl-CoA. Now, fatty acyl-CoA needs to get into the mitochondrion where the oxidation occurs, but there's a little complication with that. The acyl-CoA won't directly go into the mitochondria. So the cell has wasted some energy, in some thought, by putting this CoA onto the acyl-CoA, and then we see it gets taken off. So in the next step of the process, starting in the cytoplasm on the left, we see that the acyl-CoA is combined with a molecule called carnitine to make an acyl-carnitine. Now, this step is essential for the movement of the acyl group into the mitochondrion, and the reason is the CoA just can't make that move across the lipid bilayer. I should also mention that we're looking at the mitochondrion here. So on the right side where it says matrix, we're talking about the inside of the mitochondrion, and of course on the left side where it says cytosol, we're talking about the cytoplasm of the cell.
In between the two, you see the yellow and blue lines that are drawn there. And those relate to two layers of membrane covering the mitochondrion. The yellow one being what's called the outer layer, and the blue one, what's the inner layer. The blue one is very impermeable to a lot of substances, whereas the yellow one is fairly permeable. So you see the acylcarnitine moving into what's called the intermembrane space, the space between the yellow and the blue lines. And then when it gets there, it encounters the yellow protein there that is a transfer protein that physically moves the acylcarnitine into the mitochondrial matrix, as you see. Now, once it gets in the mitochondrion, then the reverse of all this process actually happens. The CoA that was floating around the mitochondrion is now attached to the acyl group, and the carnitine is released. The carnitine ultimately makes it back out into the cytoplasm, and the process continues. So what we've done here is what we describe as a shuttle. The shuttle has carried the acyl group from the cytoplasm into the mitochondrion. We have an acyl-CoA in there, and the acyl-CoA is the target for the oxidation. Beta oxidation proceeds, now starting with that acyl-CoA. So we see now the step-by-step process whereby a fatty acid is oxidized. The oxidation in this process is shown in the first step below. We see an enzyme called acyl-CoA dehydrogenase, and it does the very first reaction on that fatty acid. You can see that all the action in fatty acid oxidation occurs between carbons 2 and 3, or the alpha and beta, depending upon how you call it. The first oxidation is the removal of electrons and protons, and those are donated to FAD to make FADH2. The result of that oxidation creates a trans-bonded fatty acid called trans-delta-2-enonyl-CoA. That name isn't very commonly used, by the way. But the important thing is it's a trans fatty acid. And I note that it's not related to trans fat. Trans fat happens or is made for other reasons. This is a normal intermediate in fatty acid degradation. Trans fat is typically made by chemical treatment of food. Now, there are three forms of this enzyme. There's a form that works on long fatty acids, typically longer than about 20 or so, a form that works on medium-sized fatty acids between about 18 and 10 carbons, and then there's a form that works on short fatty acids, typically less than 10 carbons. Now, of note, the acyl-CoA dehydrogenase that works on the medium form of the acid is a form that is absent in infants who have died from sudden infant death syndrome. Now, it's not a cause-effect, but there appears to be a relationship between the absence of this enzyme and SIDS. Now, the FADH2 that was generated in this process can be taken into oxidative phosphorylation and used to make ATP. So we're seeing that these oxidative processes are used to generate energy that the cell can use for other purposes. Now, in the second reaction of fatty acid oxidation, we see that this trans intermediate gets a water added across it. This is a very common reaction in organic chemistry and is catalyzed by the enzyme enoyl-CoA hydratase. This enoyl-CoA hydratase creates a molecule that has a hydroxyl group on the beta carbon, or carbon number 3, and that hydroxyl group is in the L configuration, as you can see here. Now, this reaction is similar to a reaction that occurs in the citric acid cycle. In fact, this reaction and the next two reactions are very similar to citric acid cycle reactions. And this allows for a oxidation that will occur in the next step. So the second oxidation of fatty acid, fatty acyl-CoAs occurs by the process that you can see here. On the bottom, you see that we have the hydroxyl fatty acid that we created on the last step. And on this step, we see that that hydroxyl group is converted into a ketone. That oxidation is catalyzed by the enzyme that you see on the screen and causes production of NADH plus H+. That is the transfer of electrons and protons, as we have seen before. Now, this reaction is similar to the malate dehydrogenase reaction of the citric acid cycle. This reaction is preparing us for the final step in which we will actually cleave the fatty acid into two pieces. The NADH, of course, like the FADH2, can be used to generate ATP and oxidative phosphorylation. Now, in the last step of the process, we see the keto of acyl-CoA that was created in the last step of the process being broken into two pieces. So the bond between carbons 2 and 3 or the alpha and beta, as you can see here, is broken. And the product of that is an acyl-CoA that has been shortened by 2 carbons and an acetyl-CoA. Now, the acetyl-CoA itself can be targeted for additional oxidation in the citric acid cycle. If we look at the oxidation, for example, of the fatty acid palmitate, which has 16 carbons, the complete oxidation of that fatty acid can generate close to 140 ATPs. So fatty acids have a lot of energy stored within inside of them. Now, the thiolase enzyme that catalyzes this reaction is interesting because obviously it's catalyzing this breakdown, but also the reverse reaction of this creates an important intermediate if the acyl-CoA only has 2 carbons. So if we have an acyl-CoA that only has 2 carbons on the right side, then we can combine that with the acetyl-CoA. Well, an acyl-CoA that contains 2 carbons is also acetyl-CoA. Now, what thiolase does is it joins together 2 molecules of acetyl-CoA in the beginning of a process of the formation of important energy molecules known as ketone bodies.
Okay, so this last part about acetyl-CoA and acyl-CoA we didn't cover. I don't want you to look into that. I just want you to understand what was happening during this process from acyl-CoA to the generation of the acetyl-CoA. Now, again, there's a lot of enzymes that has been mentioned. I'm not necessarily asking you to memorize the name of the enzymes in this process, particularly here. Just focus on the ones that I extensively described in the previous slides. Okay, so we've produced, so this is just another summary slide to the video that we have seen. It just shows you the processes, how we get from acyl-CoA into acetyl-CoA, which is particularly here. It's just written in steps there, so it will give you a better clarity of this process. So therefore, now we can have the same molecule, the acetyl-CoA, from either glucose or fatty acids, and then produce the energy in the TCA. Now, the third source of energy is the amino acids. And in this slide, you can see that some of these amino acids, they are listed here: alanine, cysteine, glycine, serine, and threonine. Those can feed into pyruvates. So they can be converted into pyruvates. And the moment we have them as pyruvates, you have to remember that they will eventually be transformed into acetyl-CoA and go into the cycle. So some of these simple amino acids will be directly feeding as a pyruvate. Some others, like these are more complex in their side chains, like phenylalanine, tyrosine, leucine, lysine, and tryptophan. Those will be deaminated and transformed into the acetyl-CoA. So they don't go to the pyruvate, then acetyl-CoA, they will directly go to the acetyl-CoA and directly to the acetyl-CoA. Now, some others will have properties to directly become, without an intermediate, into acetyl-CoA. And most importantly, arginine, histidine, glutamine, and proline, and this is a really important 2 groups that I want you to remember. These groups here, leucine, isoleucine, and tryptophan, that can be directly deaminated to form the acetyl-CoA, and arginine, histidine, glutamine, and proline that could form the glutamate as an intermediate and then directly enter the Krebs cycle at the alpha ketoglutarate level. Therefore, you could see that these simple amino acids now can feed in, and if you continue with the slide more, you will see that isoleucine, methionine, and valine could feed in succinyl-CoA. Tyrosine and phenylalanine could feed into the fumarate. Aspartate and asparagine is at the level of the oxaloacetate. So out of these, I want you to remember these two groups because they will come back later in a few of the diseases, this group here and this group here. But this is showing you that simple amino acids can feed right away into the TCA. And this session today has shown you how we go from the food that we intake into the digestion, into the processing, and end up into the generation of the ATP and the usage of the electrons with these molecules in the electron carriers. So all that is happening in the mitochondrial inner membrane, in the mitochondrial matrix, sorry, with part of it that is very depending on the mitochondrial inner membrane, which brings us to the next question, that the TCA cycle is such a signaling hub that is depending on the nutrient availability, the nutrient to be broken down and the energy being transmitted and then used as ATP, but also it could be a signaling hub because the change in the levels of these molecules could tell a lot what is going to happen in the cell or what are the molecules that can be changed up or down in the cell. So I wrote down here some of the examples. And this is a very, very interesting paper that came in Nature not very bad, like a long time ago, where it shows the relation between different metabolites of the Krebs cycle or the TCA cycle with different molecular aspects, such as stem cell function, the regulation of the body temperature, the blockage of the tumorigenesis, and in terms of the immune responses. So, for example, if we look at succinate can feed out to all these 4 different processes. And then if you remember, this intermediate between citrate and isocitrate also can feed to some of these molecules. Acetyl-CoA itself is considered one of the signaling molecule that plays a very important role in the lymphoangiogenesis, which is a process regulating the generation of the lymphatic organs. And not only this, so this also can tell you a lot about the healthiness of these mitochondria, because as much as this process is taking place within the mitochondria, it's indirectly telling you that we have enough nutrient, we have the mitochondrial integrity is there, the inner membrane and the outer membrane are there intact, and the barrier between the two is performing the function perfectly, and we have enough oxygen to perform these functions. And in other words, whenever there's a change in those, you can directly tell that either the barriers are broken, so inner or outer membrane has lost its integrity, some of the electron transport carriers are not performing the function, we have loss of oxygen or other causes. So the TCA cycle is not just an end cycle for generating ATP, but it also feeds back to the cell function. So again, if now you go back and watch this video that we saw in the beginning, now you have a different perspective of FADH and H2 and NADH plus, and how the energy is being generated, okay? The second thing is that some of these TCA regulated metabolites could directly play a role in particular processes that we have already discussed before. You remember we discussed that chromatin condensation and expression of genes is highly dependent on how the chromatin is condensated and how the proteins are interacting with the chromatin, the DNA molecules, right? And particularly, remember that acetylation is mostly known to induce the gene expression, while methylation is mostly known to block the gene expressions. So here, in this paper, there's a description of the alpha ketoglutarate being, as well as the acetyl-CoA, being signaling molecules that determine the activity of the histone demethylation. So in other words, the more or less of those being accumulated, this can feed back to the transcriptional level. So you can block, turn on, or turn off certain genes, just depending on the levels of the alpha ketoglutarate in the cells. And this is probably how the cell will recognize if we have enough energy and nutrients in order to turn on some genes or turn off some genes. So in this particular example, the alpha ketoglutarate is an essential cofactor for the histone demethylases. And when you say demethylases, so this means you are demethylating those histones. In other words, you are removing the methyl groups, and therefore you're making this more accessible for the gene expression. And another example is written here, the succinate, when it accumulates, it works as an antagonist for this reaction. So the more succinate being generated and fumarate, the more that this will be blocking the process of the demethylation or the gene expression. And probably you have to ask why succinate accumulation caused that. Because succinate is at the level where it's in the middle of the cell cycle, of the TCA cycle. Therefore, there must be a reason why it's not being metabolized into fumarate and back to the oxaloacetate, right? And this could tell that there's a reason that the cycle is not continuing, and therefore there's an accumulation of this molecule. And that has to signal toward the mitochondria, from the mitochondria to other organelles like the nucleus, in order to turn off certain genes. Okay, so this is another example why we were really stressing and to understand and study the cell cycle. And this slide from the module 4 was probably you can go back and refresh your memory about the histone methylation and demethylations and its relation between turning on and off the genes. Two other examples I'm going to show you, which is particularly the HIF, the hypoxia-induced factor alpha, that is normally hydroxylated by this enzyme pyrrolorhydroxylase. And once it's hydroxylated, the H1 alpha hydroxylation, meaning you're adding just the OH groups. Now, when it's hydroxylated, it will be degraded in the proteasome. Okay, now in hypoxia, in normal conditions, this process takes place. And hypoxia-induced factor is, in fact, a sensor for the loss of the oxygen. So whenever there's a hypoxic condition where you don't have oxygen, this process is blocked. And therefore, if this process is blocked, H1 alpha, HIF1 alpha, is not going to be degraded. It's going to be accumulated. Now, what would cause H1 HIF alpha to be accumulated? It's the side products of the loss of oxygen. Like, in other words, succinate, fumarate, and L2HG, these are the products when you don't have enough oxygen to proceed with the Krebs cycle. And it's when H2O2, which is a reactive oxygen species, has not been removed. Therefore, when you have a situation like this in a hypoxic condition, this will block the degradation of the H1 HIF alpha, and then HIF alpha can now accumulate, goes to the nucleus, and turn on multiple genes. So this is also a feedback that can show you the Krebs cycle within the mitochondria can feed back to tell you what is the level of the oxygen in the cell and whether the level of the oxygen in the cell is low or high, depending on these metabolites that are being accumulated there. And finally, this probably has already been mentioned before, but this is a different example where within mitochondria, we have other examples where increase of hypoxia or acidity, meaning loss of oxygen or increase of the acidic metabolites, could cause the generation of the alpha ketoglycerate, which we have seen in the first slide here, the alpha ketoglycerate. And now this can feed back into the cell by directly affecting the DNA hypermethylation. Okay, because this is a process where you can tell it might happen in certain types of cell, and this is very well described in the T regulatory cells. So I want you to relate these three different slides that I'm showing you here to the fact that TCA is not simply a cycle to generate energy, it's also a signaling process where accumulation of these molecules, and I want you to remember that for example, here, I want you to remember that the accumulation of the alpha ketoglycerate leads to the generation of this L2HG buildup. And this is the intermediate that causes the process of the DNA hypermethylation. Okay? Those are examples at the molecular mechanisms that certain aspects of cell related transcription or other functions has been changed, right? If we move a little bit further and we zoom out, then we will go back to the adaptation process. So these could be simple changes that the cell is going to take as an adaptation process. In order to see if this continues, probably the cell has to send more signals that it needs to be cleaned up. And the cleanup would be probably the cell is going to die. So the cell has to face the adaptation in different ways. We discussed some of those by the atrophy, hypertrophy, metaplasia, and such, before we consider that this is an injury. And now, probably if such signaling, any of this signaling that I just showed in the last three slides continues, then this process is going to be very complicated, and the cell has to take a decision whether it's irreversible or an irreversible. Now, if it's reversible, the ATP is lost, but it can still be recovered. The mitochondria has a swell, but the complexes are still in place in the inner membrane. The ribosomes had detached, but there's a possibility that these ribosomes will reattach to the messenger RNA and recover the process of protein synthesis. And there's an autophagy that could take place within the smaller vacuoles within the cell. But if it becomes irreversible, this means the mitochondria has been broken down into smaller vacuolization, and therefore the inner membrane of the mitochondria has been disrupted, and electron chain carriers, transport carriers, so complex 1, 2, 3, 4, and the ATP synthase are not in place, and therefore the proton gradient has been lost. And this is most of the time is accompanied by an influx of calcium. And this means the cell has went into an irreversible point, which means the cell death. Okay, so with this slide, I think we just covered all these 6 components between ATP depletion, mitochondrial damage, misfolding we have described before, membrane damage we have described before, reactive oxygen species. So all the 6 components of the molecular aspects that could cause the cell injury has been covered, which means in the next round of sessions, as I mentioned, we will start discussing causes of cell death and their relation to diseases, and we will start to describe disease by disease, or maybe a couple of diseases in each session, and we will feed back into these mechanisms as we go. Okay, I will leave the calcium injury for the next, the beginning of the next class. I think this class was very heavy. Please digest it till next week. By the time, if you have any questions, let me know. If there's anything to be clarified more, I will be more than happy to go through these slides. And this will be the last thing I will describe today. Okay, so then let me just stop the recording and